Did you know that malaria still affects almost 50% of the globe, mostly in Africa and South America, killing thousands of children every single day? This disease is known already for thousands of years, and we still don't have any effective vaccine for it. This disease is transmitted by a female Anopheles mosquito that transmits an intracellular parasite named Plasmodium that has 5 species that infects humans, and the deadliest of them all is Plasmodium falciparum, which we are studying in the lab. This parasite has a fascinating complex life cycle. Infection begins with the blood meal of the mosquito, transmitting the parasite to the human body, enters to the hepatocytes in the liver, and stays there, proliferates there for about 14 days, which the patient doesn't feel anything at this point. Then this parasite bursts into the bloodstream, enters to another host cell, the red blood cell, and this is the stage that we are studying in the lab. This is the stage that the symptoms begins: chills, fever, severe anemia, and even coma and death. In 48 hours, the parasite invade a red blood cell, develop and divide to 20 to 30 daughter cells. Then the parasite will burst into this another red blood cells and infect another red blood cell. And if we will know how to stop any of these steps, we will stop malaria transmission. This parasite is encapsulated by multiple membranes. And you can imagine that for such a complex journey, it had to develop means in order to coordinate its actions. So this is what we are studying in the lab, the communication of the parasite. We and others already showed that the parasite knows how to communicate through extracellular vesicles with itself and with its host cells. These extracellular vesicles are bubble-like organelles that contains protein, RNA, and DNA. But what about other communication systems? We know that bacteria and other microbes use small molecules and metabolites in order to communicate with themselves. But this field is still unknown in malaria. So we decided to open this question in our lab. Does malaria parasite use metabolites in order to communicate with itself or with its hosts? In order to answer this question, we needed to establish a complex pipeline. Briefly, we are collecting media from the parasite, passing through several separation steps, and eventually send it to metabolomic analysis. From the metabolomic analysis, we got a lengthy list of molecules. But one molecule drew our attention. It was pipecolic acid. It drew our attention because it was highly abundant in the parasitic culture, and it was absent in the control, the uninfected red blood cells. At this point, we knew nothing about this molecule. But let me tell you what we've learned. Pipecolic acid is a non-proteogenic amino acid, and it is an intermediate metabolite from lysine degradation pathway. And what was interesting to find out that in malaria patients, in severe malaria patients, which patients enters to a coma, they have this pipecolic acid in high levels in their blood. And if they do, it's actually bad news for the family. So obviously, we understood that it is important enough molecule to study because it bridges the gap with the clinics. So first thing that we wanted to ask is when the parasite secreted. Does it happen through all of the 48-hour cycle, or does it have a short window of time? We collected media from different stages of the parasite and sent it to targeted metabolomics. And surprisingly, we found out that the parasite secreted at the later stage of the cycle, just before it invades new red blood cells. But what is the motivation of the parasite to secrete this molecule? As I told you before, the most important host cell of the parasite is the red blood cell. So we decided to pre-treat red blood cells with this pipecolic acid. Then we introduce the parasite to these treated cells and monitor their growth. And remarkably, we found out that the parasite really liked these treated cells and grew in it much better. But why? we know that it is easier for the parasite to invade softer membranes. Therefore, we decided to measure the mechanical properties of these cells using an atomic force microscopy. It's a microscope that can pinch the cell and measure the stiffness of the surface. So here I'm showing you that we managed to scan red blood cells, and we have multiple colors here. The white color represents stiffer cells, and the brown color represents softer cells. And when we treated red blood cells with this pipecolic acid, we saw much more brown cells, meaning pipecolic acid significantly softened these cells. Then we wanted to ask, what is happening beneath the membrane? We know that the cytoskeleton is the one that supports the membrane. Therefore, we asked, can we scan also using atomic force microscopy the cytoskeleton? And the answer was yes. We managed to expose the cell, and you can see here in non-treated cells we have organized cytoskeleton. But when we treated red blood cells with pipecolic acid, we see disruption in the cytoskeleton and large gaps, meaning pipecolic acid softened the cells and disrupting the cytoskeleton. Now we wanted to dig even deeper and ask how pipecolic acid softened these cells. And we needed to be creative at this point because you need to understand that red blood cells doesn't have any organelles. So we decided to look at lipids of the red blood cell. We designed the liposomes with the lipids that is abundant in the red blood cell. And using these liposomes, we could approach electron paramagnetic resonance. It's a machine that can measure movement of unpaired electrons. If an unpaired electron, is in a rigid membrane, we will get a signature from this machine that looks more like this. But if an electron has place to move in a mobile membrane, we will have a signature that looks more like this. And this is what we did. We designed a liposome with unpaired electrons, and we treated these liposomes with pipecolic acid and measured it in the EPR. And we were very happy to see that pipecolic acid managed to shift this signature, meaning pipecolic acid interacts with the lipids, making it more mobile, and maybe this is how it softened the cells. Now that we discovered the phenotype of pipecolic acid, we wanted to know, can we inhibit pipecolic acid in order to help these poor cases of severe malaria? In order to do so, we needed to find some enzyme that is part of pipecolic acid formation, and it's still unknown in malaria. We found this enzyme that has homology in a different organelle in bacteria, and we decided to try and manipulate it, tag it by CRISPR-Cas9, and to see what will happen. It took us only 2 years to clone this parasite, and we managed to transform it to the parasite. And then we treated the culture with a drug to inhibit this enzyme. We collected media from the culture that we treated with drug and sent it to targeted metabolomics. And it was very exciting to see that we managed to reduce pipecolic acid in 40%. So maybe with this gene and with other genes that we will find, we will manage to reduce pipecolic acid secretion and help these cases of severe malaria. So I hope with all this data, I managed to convince you that malaria indeed interacts with small molecules, in this case pipecolic acid. And we opened a new field in malaria research. So I want to thank to all my lab members and the researchers that helped me with this project, and thank you for listening.